Your body is running on the metabolism of an ancient bacterium that got trapped inside your ancestors over 1.5 billion years ago, and it still has its own separate DNA to prove it. That's not a theory. That's happening inside every single one of your cells right now. Fact number 1. Mitochondria have their own completely separate DNA, and it looks nothing like yours. Here's where things get immediately strange. Open up any human cell and you'll find two completely different sets of genetic code coexisting inside it. One set lives in the nucleus, that's your standard 23 pairs of chromosomes, the DNA you usually hear about. But tucked inside every mitochondrion is a second, totally independent genome. Scientists call it mtDNA, and it is nothing like nuclear DNA. While your nuclear DNA is wound into elegant linear chromosomes, mitochondrial DNA is a small, circular loop, exactly like the DNA found in bacteria today. We're talking about 16,569 base pairs arranged in a ring, encoding just 37 genes. Your nuclear genome has roughly 20,000 genes. Mitochondria have 37. And yet without those 37, you would be dead in seconds. The circularity isn't a quirk. It's a fossil. It's direct molecular evidence that mitochondria descend from free-living bacteria that were engulfed, but never digested, by a host cell more than a billion years ago. Every time a scientist sequences mitochondrial DNA, they are essentially reading a letter written by a bacterium that chose, or was forced, to never leave. Visual note: show a side-by-side infographic comparing the circular mtDNA loop against linear nuclear chromosomes, with base pair counts labeled clearly. Fact number 2. Every mitochondrion in your body descended from a bacterium that invaded a cell 1.45 billion years ago. This fact has a name. It's called the endosymbiotic theory, and it was considered so outrageous when Lynn Margulis formally proposed it in 1967 that the paper was rejected by at least 15 scientific journals before finally being published. Margulis argued that mitochondria were once free-living alpha proteobacteria, specifically related to a group called rickettsials, that were absorbed by a larger archaeal host cell in what became the most consequential biological merger in the history of life on Earth. The host cell got a turbocharged energy supply. The bacterium got protection and resources. Neither became the same again. Today, the closest living bacterial relatives of mitochondria are organisms like Rickettsia prowazekii, the bacterium that causes typhus. Think about that. The engine powering your heartbeat right now is the evolutionary descendant of a typhus cousin. Scientists confirmed this in 1998 when researchers at Uppsala University in Sweden sequenced the complete genome of Rickettsia prowazekii and found its gene content strikingly similar to mitochondrial genomes, publishing the results in the journal Nature on November 12, 1998. Margulis didn't live to see full vindication, but she came extraordinarily close. Visual note: animate a timeline infographic from 1.45 billion years ago to present, showing the endosymbiosis event as a color-coded merged diagram. Fact number 3. You inherited every single one of your mitochondria exclusively from your mother, 0 from your father. This one rewires how you think about inheritance entirely. Standard genetics tells you that you get half your DNA from mom and half from dad. That's true for your nuclear genome. But for mitochondrial DNA? Your father contributed absolutely nothing. 0. Zilch. Here's why. Sperm cells do contain a small number of mitochondria. They need them to power the flagellum that drives them forward. But the moment a sperm fertilizes an egg, the paternal mitochondria are actively tagged for destruction by the egg's own cellular machinery. The process is called paternal mitochondrial elimination, and it's deliberate. Proteins called ubiquitin mark the sperm's mitochondria for autophagy, essentially telling the cell to eat and recycle them before they can contribute to the embryo. The result is that every mitochondrion in your body traces back through an unbroken maternal line. Your mitochondria came from your mother. Hers came from her mother. Hers from her mother. Back and back and back, through an unbroken chain of women, through every human civilization, through our pre-human ancestors, all the way to the origin of our species. This maternal-only inheritance is also what makes mitochondrial DNA one of the most powerful tools in forensic science and ancestry tracing ever developed. Visual note, display a branching maternal lineage chart styled like a family tree, color-coded to distinguish maternal versus paternal lines. Fact number 4. Scientists traced all mitochondrial DNA on Earth back to a single woman who lived 150,000 to 200,000 years ago. She has a name. Scientists call her Mitochondrial Eve. She was a real woman, a specific Homo sapiens female who lived somewhere in sub-Saharan Africa between 150,000 and 200,000 years ago, and every single human being alive on Earth today inherited their mitochondrial DNA from her. This was first calculated by geneticists Alan Wilson, Rebecca Cann, and Mark Stoneking, whose landmark paper was published in the journal Nature on January 1, 1987. Now, here's the critical nuance that makes this even more mind-blowing rather than less. Mitochondrial Eve was not the only woman alive at the time. She was not the first woman. She simply won the genetic lottery of mitochondrial inheritance. Every other female lineage from her era eventually hit a dead end, not because those women didn't have children, but because at some point in their descendant line, a generation produced only sons, and mtDNA passes only through daughters. Eve's line never hit that break. So when you look at a map of all human mitochondrial haplogroups, the major branches of human mitochondrial ancestry, they all converge on a single point. One woman. One unbroken maternal chain. Connecting 8 billion people alive today. Visual note, show a world map with color-coded mitochondrial haplogroup distribution overlaid, with arrows converging to a point in sub-Saharan Africa. Fact number 5. A single human cell can contain up to 2,000 mitochondria, and they outnumber your own DNA by thousands to one. When people picture mitochondria, they usually imagine one or two little bean-shaped blobs floating in a cell. The reality is almost incomprehensibly different. The number of mitochondria per cell varies dramatically depending on that cell's energy demands. A red blood cell has 0, it shed them during development to maximize space for hemoglobin. A resting liver cell carries roughly 1,000 to 2,000 mitochondria. But here's the one that stops people cold, a single human egg cell, the oocyte, contains approximately 100,000 to 600,000 mitochondria. That is not a typo. Up to 600,000 mitochondria, packed into one cell, because the egg needs to provide enough cellular energy to power an entire embryo through its first critical divisions before the embryo develops its own mitochondrial supply chain. Now scale that across your entire body. Your body contains approximately 37 trillion cells. The average cell carries somewhere between 1,000 and 2,000 mitochondria. Conservative estimates suggest your body contains somewhere between 37 trillion and 74 trillion individual mitochondria, each carrying its own circular genome. Each a descendant of that ancient bacterium. You are not one organism. You are a superorganism hosting a civilization of ancient bacterial descendants that have been with us since before complex life existed. Visual note, use an animated infographic showing a cell cross-section with mitochondria count per cell type displayed as a bar chart: liver, heart muscle, egg cell, red blood cell. Fact number 6. Mitochondria don't just make energy, they are the ones who decide when your cells commit suicide. The powerhouse of the cell is also the executioner of the cell. This process is called mitochondria-mediated apoptosis, programmed cell death, and it is one of the most important biological mechanisms keeping you alive right now. Here's how it works. When a cell is damaged beyond repair, mutated DNA, viral infection, cancerous transformation, mitochondria receive stress signals and respond by releasing a protein called cytochrome C from their inner membrane space directly into the cell's cytoplasm. That release is essentially a death sentence. Cytochrome C triggers a cascade of proteins called caspases, which systematically dismantle the cell from the inside out, breaking it into tidy, disposable fragments that immune cells can clean up without triggering inflammation. This is not cell death by accident. This is cell death by decision. Your body destroys somewhere between 50 billion and 70 billion of its own cells every single day through apoptosis, a number confirmed by research published in Cell Biology International. Without mitochondrial apoptosis functioning correctly, cells that should die keep living. That is one of the core mechanisms of cancer. The mitochondria aren't just the powerhouse. They are the security system, the judge, and the executioner, all at once. Visual note: show a step-by-step diagram of the apoptosis pathway, mitochondrial stress signal cytochrome C release caspase cascade cell fragmentation. Use color-coded arrows on a clean white academic background. Fact number 7. Mitochondria generate your body's entire energy supply using a molecular motor that spins at 9,000 revolutions per minute. The mechanism by which mitochondria actually produce ATP, adenosine triphosphate, the energy currency your body runs on, involves a nanoscale rotary motor so elegant and so improbable that when its structure was first described, multiple scientists compared it to a machine that shouldn't exist in biology. The machine is called ATP synthase. It sits embedded in the inner mitochondrial membrane and works like a turbine. Hydrogen ions, protons, flow through it down a concentration gradient, and their movement physically rotates the central shaft of the enzyme. That rotation mechanically forces the synthesis of ATP from ADP and inorganic phosphate. The rotational speed of ATP synthase under normal cellular conditions is approximately 8,000 to 9,000 revolutions per minute. For comparison, a Formula One engine at full throttle spins at around 15,000 revolutions per minute. ATP synthase reaches over half that speed. Inside a machine you need an electron microscope to see, and it does so inside every living cell, in every organism on Earth, with extraordinary efficiency. Your body produces and recycles its own body weight in ATP every single day. On a physically active day, that number can reach twice your body weight. Every gram of that ATP passed through a spinning molecular motor inside your mitochondria. Visual note: animate an ATP synthase rotation diagram with RPM counter displayed, cross-referenced against a comparison chart showing F1 engine RPM for scale context. Fact number 8. Mitochondrial DNA mutations accumulate over your lifetime and may be the primary biological clock driving human aging. We've saved the most personally relevant fact for last, because this one is about you, right now, and how long you have. Mitochondria are the primary site of cellular energy production, which means they are also the primary site of a damaging byproduct called reactive oxygen species, free radicals. These are unstable molecules generated during ATP production that can damage proteins, membranes, and critically, DNA. Because mitochondrial DNA sits immediately adjacent to the inner membrane where this chemical storm occurs, unlike nuclear DNA, which is safely coiled in a separate compartment, mtDNA accumulates mutations at a rate approximately 10 to 17 times faster than nuclear DNA, according to research published in the Proceedings of the National Academy of Sciences. Over decades, these mutations build up. Mitochondrial function degrades. Cells produce less ATP. Tissues that depend on high energy output, heart muscle, neurons, skeletal muscle, begin to fail. This mitochondrial dysfunction is now considered by many leading biologists, including researchers at the Buck Institute for Research on Aging, to be one of the primary drivers of the aging process itself. The 2013 paper by David Sinclair and colleagues at Harvard Medical School, published in Cell, demonstrated that restoring mitochondrial function in aged mice produced measurable reversal of aging-related physical decline within just 1 week. 1 week. Your mitochondria are not just powering you. They are keeping score. And the score they're keeping is your age. Visual note: display a split infographic. Left side shows a healthy young cell's mitochondrial network in a clean diagram. Right side shows a fragmented, mutation-marked mtDNA loop representing an aged cell. Use a mutation accumulation timeline bar beneath both. And there it is, 8 facts about the most extraordinary organelle in biology, an ancient bacterial immigrant that generates your energy, controls your cell death, carries your maternal ancestry back 200,000 years, and may ultimately determine how long you live. The mitochondria was never just the powerhouse of the cell. It was always the most important organism you never knew you were carrying. If any of these facts genuinely surprised you, and I hope every single one of them did, hit subscribe right now. Subscribe to Moonlit Minds for daily mind-blowing facts that will change how you see the world.